I'm Sophia,
And I'm Kwame.
and we are going to be teaching you about HIV1 reverse transcriptase. But before we do that, we should probably take a quick look at what HIV is. HIV has been very historically and medically important because it's the human immunodeficiency virus that's responsible for the AIDS epidemic. It is a retrovirus, which means that once it gets into the cell, it is going to integrate its genetic material into the host genome. But the thing is, the central dogma of biology, as we learned in class the other day, is DNA goes to RNA, goes to protein, which would be totally fine if HIV was a DNA virus, but it's not, it's RNA. So basically, HIV is spitting in the face of the central dogma by saying, no, actually, I want to go from RNA to DNA, and then back to RNA, and then to protein. But the cell doesn't have the machinery to do that. So the virus has to bring it along, you can see it here, inside the capsid if it actually wants to get anything done. And this is a blown up version, as opposed to a yellow dot, of what reverse transcriptase looks like.
This is the primary structure. The most important part, the active site, is the thing that's most conserved is this YMDD. These residues right here are the active sites for the polymerase, and it has to be conserved. Like every time the protein sort of replicates itself, every time it makes new copies of itself, these amino acid groups stay the same. That does not change. All the other ones can change, and it leads to the protein's resistance against most of the medicine that we come up with. But these four amino acids stay the same.
And these colors that you see on this primary structure correspond with the colors here on this secondary structure.
And as you can see, the secondary structure is dominated by alpha helices. All these rods you see and the connections, every single rod is an alpha helix. I think there's like 13 alpha helix and connections.
And actually that secondary structure was really only looking at this domain up here, because that's the one that's really important. HIV reverse transcriptase is a heterodimer with two subunits. That's P66 up here and P51 down there. P51 is 440 residues long and P66 is 560 residues long. P51 is mainly for structure and stabilization, but the heavy lifting of the enzymatic activity occurs in P66.
You can tell there are two domains. There are two major domains for the P66 subunit. As Sophia mentioned, that is the subunit in which catalytic activity occurs. And the first domain is the polymerase domain, and the second is the RNase domain. Which two domains for the enzyme means that it has two active sites, meaning that it catalyzes two different reactions. The polymerase domain is sometimes compared to the human right hand. It has fingers, thumbs, a palm, and connection. The polymerase active site, as I was telling you about, these two amino acid residues, aspartic acid 185 and 186, are a part of the YMDD structure that I was telling you about is highly conserved. The reason why it's highly conserved is because it's a part of the active site for the polymerase. The aspartic acid residues for the active site sort of coordinate this magnesium ion, and the magnesium ion is 2+, and it coordinates with the phosphate group on the nucleic acid, and since it's 2+, it can coordinate with two phosphate minuses, two O minuses on the phosphates.
And if you look at this diagram, you can actually see the aspartic acid 110 and 185. You can see them here in this complex.
And this is the tyrosine that was a part of the conserved region. And this is the like all of this is basically really important to the enzyme. They basically position the nucleic acid in a way that the catalytic activity can happen. The RNA's active site has more sort of residues because they coordinate two Mg2 plus metals. And basically, like for the polymerase domain, this one, the Mg ion, the Mg metals are basically for like stabilization. They position the nucleic acid in a position where the catalytic activity can occur. The mechanism is actually really interesting. This is the mechanism for the polymerase, and the mechanism for both of them are non-catalytic metal ion catalysis. The polymerase domain has it needs to take on many conformations. It's compared, like I said, it's compared to a human right hand, and basically what happens first is that in the presence of nucleic acid, the thumb, this part moves up and it binds to DNTP. When it binds to DNTP, the fingers close down on the nucleic acid, and the fingers closing down is actually the rate determining step. It's the slowest step in the whole catalysis. After the DNTP is used, the fingers open back up and the nucleic acid is allowed to move relative to the polymerase, the domain. And it's called translocation, the moving of the nucleic acid. And then catalysis can happen on another part of the nucleic acid. The RNase mechanism is sort of the point of the RNase domain is to degrade the nucleic acid. So the product of the polymerase domain is an RNA-DNA duplex. And the RNase basically degrades the RNA part of the duplex so that the DNA can go back so that it can become double-stranded. And then after it becomes double-stranded, it can be integrated into the host genome. And so what happens is that the Mg ions sort of position the nucleic acid, the RNA-DNA duplex, in such a way that the RNase domain can hydrolyze the middle of the bond, cleaving the RNA from the DNA.
All right. Now, a lot of things are known about the structure and function of reverse transcriptase. It's really well studied, especially in the 80s and 90s, because that was when it really broke out into the human population. In 1997, Birgitta Worrell, I think that's how you pronounce her name, and her colleagues in 1997 did a study on mutating a certain region in P66 that was thought to grip the DNA so that the primer could be put on it. And then, because without the primer, DNA replication is absolutely not going to happen. And so, in order to do this, they picked some sites that they thought might be crucial. There's a tryptophan, 229, methionine 230, glycine 231, and tyrosine 232. What they did is they mutated the primary sequence so that all of these were replaced with alanine, obviously one per group. And then the results were gathered by a standard assay and gel filtration, chromatography. And what they ended up finding was that they could track how much product was being produced. And what they found, as you see in this table here, is that the enzymatic activity compared to the unmutated form was way, way lower, especially for this tryptophan, wow, 229 substitution, where you only get 0.1% of wild type activity. And this line here is showing the tryptophan 229, and here you have DTTP concentration. That is basically what is used in vitro to put together, it's used in vitro to put together DNA strands. That's what they're adding. So they're adding substrate, basically. And then this is rate. And as you can see, we see those nice Vmaxes up here. But these are all, these two mutants especially 229, showed seriously lower activity.
Okay, and if you look at this graph and compare the KDs, you can see that since Vmax for this tryptophan, the mutation of tryptophan is so low, KD is right here. It's not even one. Like it's so low compared to the KD of the original.
And this is basically showing the main sites that were mutated. This is the tryptophan, which it has these ring structures here that alanine does not have. Alanine is much more simple. And not to make light of it, but apparently that is a big deal for the primer being able to get onto the DNA and start the replication process.
As you can see, the wild type has the most affinity for the ligand, which is nucleic acid. And as we know, the lower the KD, the higher the affinity for the ligand, for the enzyme. And here is the tryptophan one. Which one is the tryptophan?
Yes.
Yeah, the tryptophan one. And as you can see, KD is really high. I don't really understand why this KD is higher when the graph shows that the Vmax for the tryptophan was the lowest, which would mean that it would have to have the highest KD. But I guess that's their data. So
Science, you get what you get. Now, because HIV is such a big deal, inhibiting reverse transcriptase has been something that the medical community has been working on for a long time. And they chose reverse transcriptase as a target for drugs because the virus has it, but the cell doesn't. So you can do whatever you want to reverse transcriptase, and it hopefully won't hurt your own cells. There are two mechanisms of inhibition. One requires nucleoside analogs, and the other requires non-nucleoside inhibitors, and both have drug applications. Actually, this molecule that you're seeing here is a nucleoside analog. This is AZT. Nucleoside analog inhibition is basically competitive inhibition. What you're doing is you are inserting what is essentially a junk nucleotide in here to cause chain termination. So your DNA is being synthesized quick as you please, and then this nucleotide will get inserted, but then after that, nothing else can be put on that end, and then the DNA will dissociate from this enzyme. And this happens chemically because here on this 3', there should be a hydroxyl group down there here, which would allow for the chain to continue, but instead we have 3 nitrogens double bonded to each other, which nothing is getting added to that, and that is not going to be broken anytime soon.
The non-nucleoside inhibitors inhibit non-competitively. Basically, all of these drugs are really hydrophobic, and there is a hydrophobic pocket right here. 10 angstroms away from the thumb. And what these drugs do is that they basically stabilize the open thumb configuration. And for the nucleoside to translocate, the thumb needs to sort of be, and for catalysis to happen, the thumb needs to close down. These drugs make it so that this can't happen, and so then the polymerase catalysis cannot happen.
And as you can see, we have a lot of drugs of this type, and they all have these very hydrophobic ring things. But the thing is, even though we have so many, we still have a hard time attacking reverse transcriptase, because this entire mechanism that we talked about of how reverse transcriptase works, it doesn't come with spell check, essentially. It makes mistakes. Mutations in the HIV genome happen a lot. So by the time you've been on one of these drugs for several months, the genome of the virus has mutated to work around the drug. So that's why you usually end up giving HIV patients drug cocktails and switching them up every now and again. And that's why this mechanism is still a really hot topic, because the more we have to fight HIV, the better. These are our literature references, and these are our image references. If you have questions, which I'm sure you will, you can leave them down in the comments. Thank you very much.
All right, thank you.